Hello and welcome to the Natural Phenomena Sex Differences module of the ENIGMA-U course. My name is Dan, I'm a postdoc, and I'll be walking you through this topic today. This topic is a lot more controversial and hotly debated than you might think upon initially considering the title of the talk, Sex Differences in Brain Structure, in the context of brain imaging. Can we use brain imaging to differentiate between the sexes? There are, just as of last year, there have been studies published in prominent journals arguing that we cannot, and there's no useful differences between the brains of the sexes. And there are studies arguing that there are, and it's very important to look at those differences. There's a long history as to why we study sex differences in the brain, and of course, there's a history of sexism in science, and women have been underrepresented in studies in science in general for many years. This is a brief overview, and I will be walking you through the history and what we are looking at now. So as we get started, let's start with the definition of a sex difference. Scientists use the sex determination of biological sex being sex chromosomes, XY and XX, and gonadal phenotype, testes and ovaries. This is not binary, of course. Intersex people make up about 1% of the population. And this is not related to the socially defined and malleable concept of gender. But this definition allows scientists to perform empirical studies with a tangible definition that represents about 99% of the population. I'm referencing a paper by Dicasian, Guma-Liu, and Raznahan, 2022. I referenced that paper several times throughout this talk. It's a review of modern brain imaging studies of sex differences, and I recommend you take a look at that paper if you're interested. So let's talk a little bit about the history and motivation of why we study sex differences in the brain. Briefly, there's clinical evidence that many disorders are more prevalent in one sex than the other. Men and women differ in healthy aging trends, as reported in recent studies. Sex differences are regularly found in large neuroimaging studies. And of course, there's underlying biology and mechanisms to differentiate brain structure, be it genetic, evolutionary, or endocrine, or environmental or sociological influences. Speaking on the clinical evidence, a number of disorders and diseases are known to be different in prevalence in the sexes. This includes major depression, anxiety, substance abuse disorders, psychosis, PTSD, and neurodegenerative diseases such as Alzheimer's, MS, or Parkinson's. So it really runs the whole gamut. Science knows alarmingly little as to why this is, and it's been understudied for many years. Recently, the ENIGMA Lifespan group published a paper of brain structure across the lifespan and found a number of differences between the sexes at different ages, as is displayed in this graph here on the right. One of the strongest findings was in middle age, where the slope between cortical thickness and sex was steeper in males, meaning that at middle age, the brains of males, the cortical thickness of the brains in males was dropping off earlier in age. And this may agree with previous findings of greater cognitive reserve in aging females. Moving on to underlying biology and mechanisms of differences in brain structure. Of course, there are genetic, evolutionary, and endocrine examples as to why we would study sex differences in general. Of course, naturally, the Y chromosome determines concentrations in testosterone and estrogen during development, being the SRY gene on the Y chromosome leads to divergence or differentiation between testes or ovaries, which leads to divergence in concentrations of testosterone or estrogen, which has, of course, downstream effects on development of different organs and brain structures. In one study here by Xu et al., um, they studied the brains of mice longitudinally, and they found several regions where regions that are known to be larger in males develop earlier. And here you can see a heat map of these regions, and they include the cerebellum, medulla, midbrain, pons, thalamus, hippocampus, amygdala, and so on. I won't read the whole slide to you. Further, there are physiological differences in body anatomy. There's reliable differences between the sexes in adipose tissue, the liver, immune systems, and kidneys. And the difference in immune response is such that it's led to recommendations by clinicians to adjust concentrations of vaccines to target the different differences in concentrations of antigens in males and females, particularly in anti-cancer clinical trials. And of course, it's easy to consider environmental or sociological influences on differences in the sexes. Men and women experience different environments in the world. A notable example is pregnancy. XY males do not experience pregnancy. And there have been a number of studies showing that pregnancy leads to long-lasting changes in human brain structure. And there's a paper here you can check out if you're interested in that. And one thing I can attest to is that fatherhood also seems to influence brain structure, and there is a paper describing that phenomenon, thought to be caused by the hormone oxytocin. And of course, there are gender biases and gendered experiences in our society that could cause differences in brain development or influences in brain structure. Moving on to part 3, can structural MRI find sex differences in brain anatomy? As I mentioned in the beginning, there are a number of large studies reporting differences in regional brain anatomy between the sexes using structural MRI. Structural MRI currently is the best way to study lots of people. This is because the technology is such that with a large enough sample size, we should be able to reliably find differences between groups that we're comparing statistically. And we can also control for covariates using statistics. 3 studies I'll be talking about are from the UK Biobank, the Human Connectome Project, and the Study of Health in Pomerania. And you can see this sample sizes of each of those studies here. This is a figure from the Decasian paper I mentioned earlier, showing each of these 3 studies. They're looking here at differences between the sexes in brain volume across the regions of the brain. And you can see the cool colors represent larger volumes in females, and the warm colors represent males greater than females. In true meta-analysis fashion, they took it a step further and generated a composite or a complement figure showing where significant sex differences across the 3 studies occurred in the same direction, in the same region. And you can see that here, there's quite a lot of reliability across the 3 studies, or reproducibility, I should say. Finally, in figure E, these purple regions are areas of inconsistencies across the studies and direction. So a lot smaller on the eyeball test, but worth considering and could be interesting to look at why that may be. Part 4, moving from structural MRI to advanced MRI. Advanced MRI goes beyond anatomical structure. This is, I'm referring to here, diffusion MRI, looking at white matter tracts in the brain. And from that, you can do connectome analysis. And in this ENIGMA-U course, I'm certain that this is covered in detail in another module, so I won't go further here on the methods, but I will report the findings of one study with a large sample, 949 children and young adults age 8 to 22, looking at sex differences in the connectome of the human brain. And they found that males generally show higher white matter connectivity, except for females have higher white matter in the corpus callosum. The corpus callosum being the bridge between the two hemispheres, facilitating communication between the two hemispheres. And this finding suggests that women's brains appear to be optimized for interhemispheric communication. And this is a developmental divergence that occurs around puberty. Moving on to part 5, methods and statistical considerations, which is what I will leave you with to think about today. So in these aforementioned studies and many large studies of brain structure, a common thing that we do is adjusting for height and cranial size. Subcortical volume is highly correlated with cranial volume. And we want to know, is there actually differences between males and females of the same brain size, same age, and whose data was collected under the same technical parameters? Is that even possible? And you have to think about exploratory versus hypothesis-driven research as well. And a final comment on reproducibility. This paper by Elliott et al. Titled "Dump the Dimorphism: Comprehensive Syntheses of Human Brain Studies Reveal Few Male-Female Differences Beyond Size," published in 2021. The Dicasian paper compares this study with that of Williams et al., saying sex differences in the brain are not dependent on differences in body size. I don't know what this word is here, but you can look up that paper. So these are two arguing papers, one claiming that there are no differences in male and female brains beyond size, and one claiming that they are. The Dicasian paper did some meta-analysis looking at the Elliott paper, a meta-analysis of 30,000 participants, which found few differences. But there's something of note that they did in the methodological correction for brain volumes, so methodological heterogeneity across studies. So what I was referring to earlier with controlling for brain volume to cranial size, if you're doing this on subcortical volumes, such as the amygdala, for example, there's a phenomenon known as proportionalization and isometric ratio scaling. So there are hypoallometric brain structures that do not, as the size of the brain increases, they don't increase on a one-to-one scale, a linear scale. So if you control equally across the sexes, this can lead to artificial sex bias. If you want to learn more about that, and I suggest you do, you can find this in the Dicasian paper describing this in detail. So here is my references from today's talk. This can serve also as a bit of a reading list. I definitely recommend starting with the Dicasian paper and moving on. And you can also read the argument that there are no differences in this Elliott paper here. That's what I'll leave you with today. If you have any questions, I would love to talk to you. And if I said anything wrong or misrepresented any of the studies, please let me know. I would love to fix that. And yeah, thank you and good luck in the rest of your studies.